Assalamu alaikum everyone. Information box ticket lifestyle. Biotechnology. Unit 2. Fermentation Technology. Fermentation. Fermentation can be defined as the alteration or production of products with the help of microorganisms. According to microbiologists, the term fermentation is used to describe any process for the production of products by means of mass culture of a microorganism. The term fermentation is derived from the Greek word fervire, to boil, describing the action of yeast on extracts of yeast or malted grain. The boiling appearance is due to the production of CO2 bubbles during the anaerobic catabolism of sugars. It has been used to conserve and alter food and feed since ancient times. Fermentation Technology. Fermentation technology is the use of microorganisms to produce food, biomass, pharmaceuticals, and alcoholic beverages on a large-scale industrial basis. Several species of microbes belonging to following categories are used in fermentation technology. As you can see from this table, category prokaryotes are of two types, unicellular and multicellular. Unicellular are bacteria and cyanobacteria, multicellular are also cyanobacteria. Likewise, eukaryotes also has unicellular and multicellular. Unicellular are yeast and algae, and multicellular includes fungi and algae. Microbes are biocatalysts. An industrial microorganism must: Number 1. Produce the product of interest in high yield. Number 2. Grow rapidly on inexpensive culture media in bulk quantity. Number 3. Be amenable to genetic manipulation like mutation, genetically engineered yet stable. And number 4. Be non-pathogenic, meaning they should not cause any reaction to the person who is using the product. The range of fermentation process. There are 5 important groups of commercially important fermentations. Number 1. Those that produce microbial cells or biomass as product. Number 2. Those that produce microbial enzymes. Number 3. Those that produce microbial metabolites. Number 4. Those that produce recombinant products. Number 5. Those that modify a compound which is added to the fermentation process, the transformation process. Number 1. Microbial biomass production. The commercial production of microbial biomass can be divided into 2 major processes: the production of baker yeast, the production of microbial cells for human or animal food, single-cell protein. Microbial cell biomass are grown commercially as continuous culture on a large scale. Single-cell protein. The microbial cells including algae, bacteria, yeast, molds, and mushrooms are dried and used as a good source of complete protein called single-cell protein, which serves as human food or animal feed. The incubation of wheat flour for preparation of tandoori roti is good example for the production of single-cell protein. The substrate used by the microbes producing the single-cell protein range from carbohydrates to hydrocarbons and petrochemicals. Other organisms uses the gases methane, ethane, propane, and butane, etc. as substrate for the fermentation. Organisms used in single-cell protein production include chlorella, Cellulomonas, Saccharomyces cerevisiae, Methylococcus capsulatus, and Aspergillus. Microbial enzymes. Microbial enzymes can be produced on a large scale by using fermentation technology. When microbes are cultivated, they secrete some enzymes into the media, and these enzymes are extracted and widely used in several industries like detergent, food processing, brewing, and pharmaceutical. They are also used for diagnostic, scientific, and analytical purposes. Some of the enzymes produced by fermentating microbes are glucose oxidase, protease glucoamylase, amylase, glucoisomerase, pectinase, cellulase, invertase, lactase, and lipase. Some thermophile bacteria produce enzymes that are thermostable and which can be used in industrial processes at high temperature. Example, glyceraldehyde 3-phosphate dehydrogenase, alcohol dehydrogenase, and restriction endonucleases, which are produced by Bacillus thermophilus. In this table, you can see what different functions these enzymes can do. But before that, don't forget to like this video, subscribe to my channel, and press the bell icon button. Microbial metabolites. During the metabolism of microbial cells, a number of compounds are produced and many are secreted out of the cell, which can be easily extracted and are very useful to man and animals. Therefore, fermentation by microbial cell is carried out on an industrial scale in order to get various metabolites. The metabolites produced by the microbes can be grouped into two categories: primary metabolites and secondary metabolites. Primary metabolites. Metabolites which are produced by the metabolism required for the maintenance of minimum life process of a microbes are known as primary metabolites. These are produced in abundance at early stage of growth. The phase at which these metabolites are produced is known as trophophase. Examples of primary metabolites are amino acid, nucleotides, proteins, nucleic acid, carbohydrates, lipids, etc. Many products of primary metabolites are of considerable economic importance, such as ethanol, citric acid, lactic acid, acetic acid, acetone, formic acid, ethanol, pronic acid, dihydroxyacetone, and glycerol, etc. Secondary metabolite. During stationary phase, some microbial culture synthesize compounds that are not produced during trophophase, and they do not have obvious function in cell metabolism. These compounds are known as secondary products of metabolism, and the phase in which these are produced is known as idophase. Most of the secondary metabolites are derived from the primary metabolites. The secondary metabolites include the hormone, enzyme inhibitors, antibiotics, alkaloids, toxic pigments, vitamins, etc. Primary metabolites are often released into surrounding media, whereas secondary metabolites tend to remain inside the cell and can be recovered only after lysis of the cell walls. As you can see the difference between the primary and secondary metabolites. Recombinant products. The advent of recombinant DNA technology has extended the range of fermented products. Genes from higher organisms can be introduced into microbial cells such that recipient cells are capable of producing foreign protein. Commonly used bacterial species as host for such systems include E. coli, Saccharomyces cerevisiae, and filamentous fungus. Products produced by such genetically modified organisms include interferons, insulin, human serum albumin, epidermal growth factor, etc. These processes are designed in such a way that the products are released into medium, products are degraded on very low rate, and foreign genes is expressed as maximum. As you can see in this diagram, the application of recombinant products. Transformation or modification of the substrate. The fermentating microbes have the capacity to convert an added substrate into some more valuable products. Microbes act as biocatalysts that are more specific than chemical catalysts and allow the additional removal and modification of functional group at specific sites. Microbial reactions have additional advantage over chemical reagents of operating at relatively low temperature and pressure without the need of potentially damaging heavy metal catalysts. Examples include conversion of ethanol to acetic acid, vinegar, isopropanol to acetone, glucose to gluconic acid, sorbitol to sorbus. This is used in the manufacture of vitamin C, sterols, and steroids. Among all these bioconversions, the production of steroids is the most likely applied fermentation biotechnology for the conversion of sterol into steroids, like cortisols, hydroxyl cortisols, and testosterons and testodiols, etc. Microbial growth kinetics. Microbial growth kinetics describes how the bacteria grows in a fermentator. The growth of a microbe in a fermentator can be broken down into following phases: lag phase, exponential log phase, stationary phase, dead and decline phase. As you can see in this graph, there is the lag phase, log or exponential phase, stationary phase, and dead phase. Now we will get to know what does these terms mean. Lag phase. It is the first major phase of microbial growth in a fermentation process. It is the period of adaptation of the cells to the new environment. Cells density is minimum at this phase, whereas exponential phase, also known as logarithm growth phase or log phase, cells have adjusted to their new environment. The cells are dividing at a constant rate, resulting in an exponential increase in the number of cells. Cells density is highest at this peak. Stationary phase. The third major phase of microbial growth in a fermentation process occurs when the number of cells dividing and dying is in equilibrium and can be the result of the following. Number 1. Deprivation of one or more essential growth elements, accumulation of toxic byproducts, stress associated with induction of a recombinant gene, primary metabolite production stops, secondary metabolites or non-growth associated product production may continue. Death phase, the fourth major phase of microbial growth in a fermentation process, also known as decline phase. The rate of cells dying is greater than the rate of cells dividing. The cause of death phase is usually the cells have consumed most of the nutrients in the medium and there is no enough left for sustainability. Don't forget to subscribe to my channel. Components of a typical fermentation process. Number 1. Fermentator. Number 2. Media. And number 3. Inoculum. Fermentator. Fermentation process is carried out in a container called the fermentator or bioreactor. These are used for the cultivation of microbial cell on a large scale under controlled conditions for industrial purposes. The design and nature of the fermentator varies depending upon the type of fermentation carried out. These vary from a laboratory fermentator few liter capacity to large scale industrial fermentator several hundred liter capacity. This closed metallic or glass vessel has an adequate arrangement for aeration, mixing of media by agitation, temperature control by thermostat, pH control by pH meter, anti-foaming, control of overflow, sterilization of media and vessel, cooling, and sampling, removal of sample while the fermentator is on. Here is a diagram of a fermentator with all the essential needs. Maximum cell growth and product formation can be achieved by controlling these parameters that assist in cell growth and metabolism, leading to high output of the product. The laboratory fermentators are used for optimizing culture conditions and nutritional parameters for better growth of cells and production of metabolites for conducting research studies in the laboratory. Types of fermentator. The different types of fermentator are: number 1. External recycle airlift fermentator. It is used for producing bacterial biomass with methanol as substrate. Number 2. Internal recycled air-lift fermentator, which is used for the production of yeast with oil as substrate. Number 3. Tubular tower fermentator, used for making beer, wine, vinegar, etc. Number 4. Nathan fermentator, used in brewing industry. Number 5. Stirred fermenter, used for making antibiotics. Types of culture system. The culturing of the microbial system can be achieved in different ways. The type of culture method sometimes depends on the type of microbial system or on the type of product that we expect. Depending upon the nature of required product and culture conditions, the microbial cultures are classified as number 1. Batch culture, number 2. Fed batch culture, number 3. Continuous culture. Batch culture. It consists of a limited volume of broth culture in a flask inoculated with bacterial or microbial inoculum and follows a normal growth phase. It is a closed culture system because the media contain a limited amount of nutrients and will be consumed by the growing microorganism for their growth and multiplication with the excretion of certain metabolites as products. In batch cultures, the nutrients are not renewed. The growth phase of the culture consists of an initial lag phase, a log phase or exponential growth phase, and a stationary phase. During the log phase, the consumption of the nutrients will be maximum, resulting in maximum biomass output with the excretion of the product. At the stationary phase, the rate of growth decreases and becomes zero. Number 2. Fed batch culture. The batch culture can be made into semi-continuous culture or fed batch culture by feeding it with fresh media sequentially at the end of the log phase or in the beginning of the stationary phase without removing cells. Due to this, volume of the culture will go on increasing as fresh media is added. The fermentator is designed in such a way that it can accommodate the exceeding volumes. This method can easily produce a high cell density in the culture media, which may not be possible in a batch fermentator. Industrial production of baker yeast as biomass and penicillin as secondary metabolite is an example of fed batch culture. Number 3. Continuous culture. Bacterial culture can be maintained in a state of exponential growth over long periods of time using a system called continuous culture. In continuous culture, the nutrient mediums, including the raw material, is supplied at a rate that is equal to the volume of medium with cells and products removed from the culture. The volume removed and the volume added is the same. In fact, there is no change in the net volume as well as the chemical environment of the culture. Industrial production of ethanol is sufficient by continuous culture. In continuous culture, chemostat can be used to maintain a bacterial population at a constant density, a situation that is more similar to bacterial growth in natural environment. Here is the diagram of the chemostat, which is used in the continuous culture for maintaining the nutrients in the media. Chemostat. In a chemostat, the growth chamber is connected to a reservoir of sterile media. Once the growth is initiated, fresh media is continuously supplied from the reservoir. The volume of fluid in the growth chamber is maintained at a constant level by some sort of overflow drain. The bacterial cells grow at the same rate at which the bacterial cells are removed by the overflow. Procedure of fermentation. Number 1. Depending upon the type of product required, a particular bioreactor is selected. Number 2. A suitable substrate in liquid media is added at a specific temperature, pH, and then diluted. Number 3. The organism, microbe, animal, plant cell, subcellular organ, or enzyme is added to it. Number 4. Then it is incubated at a specific temperature for a specific time. Number 5. The incubation may be either aerobic or anaerobic. Number 6. Aerobic conditions are created by bubbling oxygen through the media. Number 7. Anaerobic conditions are created by using closed vessels wherein oxygen cannot diffuse into the media and the oxygen present just above is replaced by carbon dioxide released. Number 8. After the specified time interval, the products are removed, as some of the products are toxic to the growing cell or at least inhibitory to the growth. The process of removal of the product is called downstreaming processing. Number 9. The disposal of effluent produced by the process. Design of industrial fermentation process. The fermentation process is divided into two categories. Number 1. Upstream processing. Number 2. Downstream processing. Upstream processing. Upstream processing require number 1. Pure culture of selected organism in sufficient quantity and in correct psychological state. Number 2. Carefully composed media for growth of microorganisms. Number 3. The sterilization of medium, fermentator and equipments. Number 4. A seed fermentator, a mini model fermentator to develop an inoculum to initiate the process in main fermentator. Number 5. A production fermentator, the functional large fermentator. Downstream processing. Downstream processing include drawing the culture media in steady state. Number 2. Cell separation. Number 3. Collection of cell-free supernatant. Number 4. Product purification. Number 5. Effluent treatment. Now we will talk about upstream processing. Number 1. Formulation mediator microbial culture. Like any other living system, microorganisms are also required a source of energy, carbon, nitrogen, oxygen, iron and other mineral, micronutrients and water for growth and multiplication. All these nutrients that are essential for the growth and multiplication of microbial organisms are supplied in the form of nutrients. If the nutritive components of the media are not of natural origin, such as nutritive media are known as synthetic media, which can synthesize in the laboratory following certain recipes, there are a large number of commercially available nutrient media which contains both salts and minerals. Such nutrient mediums are known as semi-synthetic media. For example, commercially available nutrient broth, trypticase soya broth, brain hard infusion broth, yeast extract, potato dextrose agar, etc. are some examples of semi-synthetic media. For factory-scale cultivation of bacteria and other microorganisms, these synthetic or semi-synthetic media are preferred, but for industrial-scale cultivation, these media are not recommended for an economical point of view. For commercial purposes, the recommended media should be cheap and available year-round. Requirements of microbial medium. The following are minimum components required in a microbial medium for cultivation of microbes in a laboratory: carbon source, nitrogen sources, microelements or trace elements, growth factors, antiforms, energy sources, buffers, and water. Number 1. Carbon source. The main purpose of carbon source is to provide energy and carbon skeleton for the synthesis of various other biological compounds. It includes sugars such as glucose, lactose, sucrose, complex polysaccharides such as starch, glycogen, cellulose, a mixture of various carbon dioxide and other compounds such as cereal grain powders, cane molasses, etc. Number 2. Nitrogen sources. The major type of nitrogen sources used in culture media are ammonium, salt, urea, animal tissue extract, amino acid mixture, and plant tissue extracts. Number 3. Microelements or trace elements. Elements required in small quantities include copper, cobalt, iron, zinc, magnesium, etc. Number 4. Growth factors. Growth factors are certain organic compounds that are essential for the growth and multiplication of cell but cannot be synthesized by the cell. Certain amino acids and vitamins are also included in this category. Number 5. Antiforms. Media rich in nutritive components such as starch, protein and other organic material and other products secreted by the growing cells can result in excessive forming while the culture media is agitated for aeration. To prevent this, the formation of foam, some anti-foaming agents are included into the media. Certain types of fatty acids such as olive oil and sunflower oil and silicones are commonly used in the cell culture as anti-foaming agents. Number 6. Energy sources. The carbon sources used in culture media such as carbohydrate, sugar, protein, lipids, etc. can work as energy sources. Number 7. Buffers. Many media are buffered at about pH 7.0 by the incorporation of calcium carbonate. Phosphates, which are a part of many media, also play an important role in buffering. Number 8. Water. Water is the base of any culture media, whether it is liquid or solid. In laboratory experiments, single distilled water or double distilled water is usually used. But in large-scale microbial cultivation for industrial purposes, then the pH and the dissolved salt present should be considered when formulating the media requirements and its concentration. Water is also required for a large number of other services in the laboratory such as cooling, heating and steaming, etc. Sterilization of media and fermentator. The media and fermentator have to be sterilized to prevent the growth of unwanted microorganisms. If the laboratory scale experiments are carried out in 100 to 1000 ml flask or in lesser volume, the media along with the culture flask can be steam sterilized with an autoclave or a pressure cooker. Steam sterilization is carried out at 120 degrees Celsius for 15 to 20 minutes under 15 psi pressure. When microbes are cultivated in a fermentator for large-scale operation, it is convenient to sterilize the fermentator as a whole with or without media. Steam is used for the sterilization of media and fermentator by passing the steam through the sterilization jacket or the coal around the fermentator. When the fermentator is sterilized without media in it, steam can be passed into this vessel through all openings, allowing it to exit very slowly. The steam pressure is held at 15 pascals for 20 to 30 minutes. Preparation of inoculum for fermentation. It is essential that the culture used to inoculate a fermentation satisfy the following criteria. Number 1. It must be in a healthy, active state, thus minimizing the length of the lag phase in the subsequent fermentation. Number 2. It must be available in sufficient large volumes to provide an inoculum of optimum size. Number 3. It must be in a suitable morphological form. Number 4. It must be free of contamination. Number 5. It must retain its product forming capacity. The process adopted to produce an inoculum meeting these criteria is called inoculum development. The quantity of inoculum normally used is between 3 to 10% of the medium volume. The cultivation process. The production strain is first cultivated in a small flask containing nutrients and agar. The flask is placed in an incubator that provides the optimum temperature for previously frozen dried cells to germinate. Once the flask is ready, the cells are transferred to a seed fermentator, which is a large tank containing previously sterilized medium. Seed fermentation allows the cells to reproduce and adapt to the environment and nutrients that will encounter later on. Following seed fermentation, the cells are transferred to a larger tank, the production fermentator, where conditions are carefully controlled. When the main fermentation is complete, the mixture of cells, nutrients and enzymes, referred to as broth, is ready for downstreaming processing. Downstream processing. Downstream processing, it is referred to as recovery and purification of fermentation product, including the recycling of salvageable compounds and the proper treatment and disposal of waste. This forms the major about 85% portion of the complete fermentation technology. First of all, the broth is conditioned, that is, the cells are aggregated and form large clumps, which makes the separation easier. The conditioning is done by heating, freezing, pH change, antibody and antigen reaction, etc. Then the conditioned broth is used for the separation of constituent, for which techniques like sedimentation, flotation, filtration, ultrafiltration, centrifugation and microfiltration are applied. Cost associated with downstream processing. The extraction and purification of fermentation products may be difficult and costly. Unfortunately, recovery cost of microbial products may vary from as low as 15% to as high as 70% of the total manufacturing cost. Atkinson and Mediator 1991 indicated the percentage of total cost being 15%. For industrial ethanol, 20 to 30%. For penicillin G, it is up to 70% for enzymes. The extraction and purification of products such as recombinant proteins and monoclonal antibodies can account for 80 to 90% of the total processing costs. Stages in the downstream process. Downstreaming process includes number 1, primary unit operation, number 2, secondary unit operations. Number 1, primary unit operation of a downstream process. Removal of insolubles. It involves the removal of cells, cell debris or other particulate matters from the fermentation broth by techniques such as fermentation, centrifugation, sedimentation, etc. Products isolation. It involves the removal of components whose properties vary remarkably from that of desired product by techniques such as solvent extraction, ultrafiltration, precipitation. Now, secondary unit operation of a downstream process. Product purification. It is done to separate contaminants closely resembling the product. It is done by affinity chromatography or ion exchange chromatography, size exclusion and reverse phase chromatography, crystallization, fractional precipitation. Product polishing. It is the final processing system which ends the packaging of the product in the form that is stable, easily transposable, and convenient. It is done by crystallization, desiccation, lyophilization, and spray drying. Depending upon the product and its intended use, polishing may include steps for sterilization, dehydration, blotting, etc. Obstacles in good recovery of the product. If a fermentation broth is analyzed at the time of harvesting, it will be discovered that the specific product may be present at a low concentration, typically 0.1 by 5 grams Tm-3 in an aqueous solution. The product may be intracellular, heat liable, and easily broken down by contaminating microorganisms. All these factors tend to increase the difficulties of product recovery. To ensure good recovery or purification, the processing equipment must be of correct type and also correct size to ensure that harvested broth can be processed within a satisfactory time limit. It should also be noted that each step or unit operation in downstream processing will involve the loss of some product. Hence, it is also important that the minimum number of operations possible are used to maximize product recovery. Criteria for correct choice of recovery process. The choice of recovery process is based on the following criteria. Number 1, intracellular or extracellular location of the product. Number 2, concentration of the product in the fermentation broth. Number 3, physical and chemical properties of the desired product. Number 4, the intended use of product. Number 5, the minimal acceptance state of purity. Number 6, the magnitude of biohazard of the product or broth, the impurities in the fermented broth, and number 8, the marketable price for the product. Downstream processing depends upon product use. Number 1, enzyme preparation for animal feed supplement, example, phytase are not purified. Number 2, enzyme for industrial use may be partially purified, example, amylase for starch industry. Number 3, enzyme for analytical use, example, glucose oxidase and pharmaceutical protein are highly purified. The recovery of extracellular product. The main objective of the first stage of the recovery of an extracellular product is the removal of large solid particles and microbial cell, usually by centrifugation or filtration. In the next step, the broth is fractionized or extracted into major fraction using ultrafiltration, reverse osmosis, absorption, ion exchange, gel filtration or affinity chromatography or precipitation. Afterwards, the product containing fraction is purified by fractional precipitation, chromatographic techniques and crystallization to obtain a product. Other products are isolated using modification of this flow stream. Finally, the finished product may require drying. The recovery of an intracellular product. Microorganisms are protected by extremely tough cell wall. In order to release their cellular content, a number of methods for cell disintegration have been developed. Methods available fall into two major categories. Physiomechanical method. Number 1, liquid shear. Number 2, solid shear. Number 3, agitation with abrasives. Number 4, freeze thawing. And number 5, ultrasonication. Chemical and biological methods. Number 1, detergents. Number 2, osmotic shock. Number 3, alkyl treatment. Number 4, enzyme treatment. Number 5, solvents. Drying of products. The drying of any product, including biological product, is often the last stage of the manufacturing process. It involves the final removal of water or other solvent from a product while ensuring there is a minimal loss in viability, activity or nutritional value. Drying is undertaken because number 1, cost of transport can be reduced. Number 2, the material is easier to handle and package. Number 3, the material can be stored more conveniently in dry state. It is important that as much water as possible is removed initially by centrifugation or in a filter press to minimize heating costs in the drying process. Dryers can be classified by the method of heat transfer to the product and the degree of agitation of the product. Number 1, tray drying. For some products, simple tray dryers are used where the product is placed on trays over which air is passed in a heated oven. Number 2, contact dryer. In contact dryer, the product is contacted with the heat surface. An example of this type of drum dryer, which may be used for more temperature stable bioproducts. A slurry is a run onto a slowly rotating steam heated drum. Evaporation takes place and the dry product is removed by a scraper blade. Number 3, spray drying. It is most likely used for drying of biological materials when the starting material is in the form of liquid or paste. The material to be dried does not come in contact with the heating surface. Instead, it is atomized into small droplets through a nozzle or by contact with a rotating disc. The droplets then fall into spiral steam of hot gas at 150 to 250 degrees Celsius. The high surface area volume ratio of the droplet result in the rapid rate of evaporation and complete drying in a few seconds. Number 4, freeze drying, also known as lyophilization or cryodesiccation, is an important operation in the production of many biological pharmaceuticals. The material is first frozen and then dried by sublimation in a high vacuum, followed by secondary drying to remove any residual moisture. Effluent treatment. Every fermentation plant utilizes raw material which are converted to a variety of products. Depending on the individual process, varying amount of range of waste materials are produced. Typical waste might include unconsumed inorganic or organic media components, microbial cells and other suspended solids, waste wash water from cleansing operation, cooling water, water containing traces of solvent, acids and alkalis, etc. Treatment and disposal of effluent. The range of effluent disposal method which can be considered as the follows. The effluent is discharged to land, river or seas in an untreated state. The effluent is removed and disposed in a landfill site or incinerated. The effluent is partially treated on site prior to the further treatment to disposal by one of the other routes. All of the effluent is sent to the sewage work for treatment. All the effluent is treated at the factory before its discharge. It may be possible to recover waste organic material as a solid and sell it as byproduct which may be used as animal feed or nutrient supplement. The marketable bioproducts helps to offset the cost of the treatment process. Histologically, it is possible to dispose of waste directly to the convenient area of land or into a nearby watercourse. This cheap and simple method of disposal is now very rarely possible, nor it is environmentally desirable. With increasing density of population and industrial expansion, together with great awareness of the damage caused by pollution, the need for treatment and controlled disposal of waste has continued to grow. Thank you for watching this video. Don't forget to subscribe to my channel to see more videos. Thank you.